welcome to this special AI IA3 session. My name is Christiane Morim, and this webinar is kindly sponsored by Future Fertility. So this is a webinar on in vitro maturation of oocytes, which is a technique that continues to gain a lot of importance within the field of assisted reproductive technologies. And in this session, we'll have a focused overview of the clinical applications of IVM, current evidence supporting its use, and emerging developments that may influence future practice. So tonight or today or this morning, wherever you are, you'll gain insight into both scientific underpinnings and the translational aspects of IVM, with particular attention to its role in optimizing patient care and expanding therapeutic possibilities. So we'll keep the intro short today, but all the extensive bios can be found on the website. And I'd like to start by introducing my first co-host and former PhD student who has gone on to great things, Dr. Emma Uni. Emma's research is mainly focusing on multidisciplinary characterization and deep exploration of human organs in order to provide a better understanding of their mechanism and develop functional and biomimetic ex vivo models.
Thank you, Christiane, for the introduction. It's a great pleasure to host this session with my PhD mentor. So Professor Christiane Andrade Amorim earned her PhD in veterinary medicine from Universidade Federal de Santa Maria in Brazil, and in 2027 transitioned to human fertility during a postdoc fellowship, focusing this time on fertility restoration in cancer patients. She's now professor and head of the research pole in physiopathology of reproduction at Université Catholique de Louvain, Brussels, with research spanning ovarian tissue preservation, photodynamic therapy, cell differentiation, and tissue engineering, my beloved subject. She also serves as Chief Scientific Officer at Profound, and she's Honorary Research Associate with the Belgian National Fund for Scientific Research, FNRS. We are joined as well by the one and only Professor Johan Smits, who is based at Free University Brussels. He became Professor of Endocrine Physiology and Reproductive Medicine in 1997 at VUB and founded the Follicle Biology (FOBI) Research Lab in 1997 as well. And he was a member of the steering committee of the Center for Reproductive Medicine at UZ Brussels, and he humbly describes himself as a researcher in oocyte in follicle biology, but he's currently implementing kappa IVM in IVF clinics. So the floor is yours.
Thank you all. Thank you all. It's a great honor to be here and a great pleasure, especially with being with many people on the panel here from Belgium, Christiane, etc. It's really a great pleasure to be here together on a topic that we both love very much. So please be interactive and please place your questions in the questions section of the portal, and please not on the chat box throughout the webinars. So furthermore, we are joined now by Jolien Fjellstad, the head of clinical embryology and scientific operations of Future Fertility. She works closely with partnering clinics to ensure smooth onboarding of innovative AI-based tools within their laboratories. And from far away, we are joined, and especially far away in the night, also by Professor Dr. Lan Vuong, who is among the pioneers of IVM in Vietnam since 1997 and achieving the country's first successful IVF pregnancy in 2006. And with almost 6,000 IVM cycles and 2,600 live births to her credit, she continues to advance the IVM technique through global collaboration, research, and training. So without further explanations, I will introduce our first speaker for today, which is Dr. Tsubasa Takahashi, who is an embryologist at the Kato Lady Clinics in Tokyo, specializing in oocyte maturation and embryo development. His recent work focuses on pronuclear morphology and its clinical significance in assisted reproductive technology. So please enjoy this presentation that was recorded earlier.
Thank you for your kind introduction and inviting me to this I3 webinar. It's an honor to be here. My name is Tsubasa Takahashi, and I'm an embryologist at Kato Ladies Clinic in Japan. Our clinic performs over 80,000 oocyte retrieval cycles in a year. And all immature oocytes obtained are routinely treated with rescue in vitro maturation, followed by intracytoplasmic sperm injection, commonly referred to as rescue IVM ICSI. Today, I'd like to share our clinical experience and discuss both the applications and challenges associated with this technique. This presentation will cover two main topics. First, the clinical application and outcomes of rescue IVM ICSI in our setting. Second, the relationship between oocyte morphology, morphokinetics, and clinical outcomes. So let's begin with the first topic. In assisted reproductive technology, mature oocytes are the primary target for retrieval. However, immature oocytes are frequently collected as well. When these immature oocytes undergo in vitro maturation, followed by intracytoplasmic sperm injection, the procedure is referred to as rescue IVM ICSI. This approach has been reported to increase the number of available embryos and contribute to healthy live births. However, incorporating rescue IVM ICSI into the workflow can make daily lab work more complicated, especially when it comes to scheduling and timing of ICSI procedures alongside other tasks. Furthermore, oocytes treated with rescue IVM ICSI generally exhibit lower fertilization and developmental rates compared to in vivo matured oocytes. In the following slides, I will present how we manage these challenges in a large-scale clinical setting and share outcome data based on our current practice. This figure shows the workflow for the clinical practice of rescue IVM ICSI in our clinic. Oocyte retrievals were performed 34 to 36 hours after ovulation induction in minimal stimulation cycles. The retrieved oocytes were immediately placed into embryo slides for individual identification and cultured in continuous culture medium after denudation and maturation assessment. ICSI was performed after confirming the first polar body extrusion. Maturation was limited until 8 a.m. on the day after oocyte retrieval, so that ICSI could be completed by noon the same day. Zygotes exhibiting 2 p.m. were considered normally fertilized. These zygotes were then transferred at the expanded blastocyst stage during single vitrified blastocyst transfer cycle. Today, I will show clinical data from 729 patients undergoing 841 cycles. A total of 2,693 oocytes were retrieved, including 617 germinal vesicle stage oocytes, 426 metaphase 1 stage oocytes, and 1,650 metaphase 2 stage oocytes. This slide shows the maturation rates of oocytes retrieved at metaphase 1 and ZB stages. Among the metaphase 1 oocytes, 79.1% reached maturation. For ZB oocytes, 82.7% underwent GVBD, and 56.3% of those subsequently matured. As a result, the overall maturation rate for ZB oocytes was 46.5%. The maturation rate of ZB oocytes was significantly lower than that of metaphase 1 oocytes. On the other hand, the normal fertilization rates after rescue IVM ICSI were comparable between metaphase 1 and ZB oocytes, although both were significantly lower than that of in vivo matured oocytes. This slide shows the developmental rates to the expanded blastocyst stage. We observe a similar pattern to that seen with normal fertilization rates. The developmental rates of normally fertilized zygotes to the expanded blastocyst stage were comparable between metaphase 1 and ZB oocytes, although both were significantly lower than those of in vivo matured oocytes. This table shows the association between maturation status at retrieval and pregnancy outcomes following single vitrified blastocyst transfer. No significant correlation was found between maturation status and pregnancy outcomes. This figure summarizes the relationship between maturation status at oocyte retrieval and the clinical outcomes discussed earlier. Immature oocytes at retrieval had lower utilization rates due to their poorer maturation rates as well as reduced normal fertilization and developmental rates compared to in vivo matured oocytes. However, pregnancy outcomes following single vitrified blastocyst transfer were comparable to those from in vivo matured oocytes. This suggests that blastocysts derived from rescue IVM ICSI may have similar pregnancy potential and could be suitable for embryo transfer. Furthermore, the rates of normal fertilization and blastocyst expansion were comparable between metaphase 2 oocytes derived from metaphase 1 and ZB stage oocytes. Therefore, in vitro matured oocytes derived from metaphase 1 stage and ZB stage may have comparable developmental competence. In the first part of this presentation, I discussed the clinical significance of rescue IVM ICSI in potentially increasing live births. However, many aspects of immature oocytes remain unclear. For example, why is the utilization rate of in vitro matured oocytes lower than that of in vivo matured ones? What factors characterize immature oocytes with low utilization rates? To address these questions, we have been conducting studies to examine the relationship between oocyte morphology, morphokinetics, and embryonic outcomes. In the next section, I will present some of the results from these studies. In the process of rescue IVM culturing for ZB stage oocytes, we observed the timing of the ZBBD, fast polar body extrusion, and cortical dynamics at the metaphase 1 stage using a time-lapse system. We then analyzed the correlations between these dynamics and normal fertilization and blastocyst expansion. This slide, this slide presents two key morphokinetic parameters we observed. First, the time intervals between ovulation induction, ZBBD, and fast polar body extrusion. And second, the presence of cytoplasmic membrane ruffling during the metaphase 1 stage. Let me show their correlation with embryonic outcomes one by one. This slide shows the results of univariate logistic regression analysis, which examined the correlation between each time interval, ovulation induction to ZBBD, and ZBBD to fast polar body extrusion, with normal fertilization and blastocyst expansion. The analysis indicated that the time interval between ovulation induction and ZBBD was negatively correlated with blastocyst expansion. Next, I would like to talk about the presence of cytoplasmic membrane ruffling during the metaphase 1 stage. Here is a video showing oocytes during the metaphase 1 stage. In this clip, you can observe the presence and absence of cytoplasmic membrane ruffling. The oocyte on the right side clearly shows membrane ruffling. So let's take a closer look. Okay. Cytoplasmic membrane ruffling was observed in 74.3% of metaphase 1 oocytes after ZBBD. The presence of membrane ruffling was negatively correlated with blastocyst expansion, but it was not correlated with oocyte maturation or normal fertilization. These findings suggest that certain dynamic events during the pre-insemination period may be associated with embryonic developmental potential. Recently, several studies have explored the use of image-based AI to evaluate oocyte quality before insemination as an alternative approach in this field. In this presentation, I will share part of our research in this area as well. In our study, we used an AI-based oocyte image assessment tool, Magenta, developed by Future Fertility. Magenta evaluates the blastocyst formation potential of an oocyte by analyzing a single pre-insemination image of a metaphased oocyte and generates a score ranging from 0.0 to 10.0. Here, we are looking at metaphased oocytes that were originally retrieved at 3 different stages: GB, metaphase 1, and metaphase 2. For each group, we assessed how the Magenta score is associated with normal fertilization and blastocyst expansion. And what we found is that regardless of the starting stage, Magenta scores were positively associated with blastocyst expansion. These findings indicate that Magenta can predict blastocyst development based on oocyte morphology, independent of the oocyte's maturation status at retrieval. From the second part of my presentation, it was suggested that morphokinetics and morphological characteristics of oocytes during the pre-insemination period may be associated with oocyte quality. This is my final slide. To briefly summarize this presentation, immature oocytes generally have lower utilization rates due to poorer maturation, fertilization, and developmental outcomes compared to in vivo matured oocytes. However, blastocysts derived from rescue IVM ICSI show similar potential to those from in vivo matured oocytes. This suggests that they remain viable candidates for embryo transfer. These findings indicate that rescue IVM ICSI can increase the number of available embryos and contribute to live births. Furthermore, morphokinetics and morphological characteristics of oocytes during the pre-insemination period may be linked to oocyte quality. Better understanding these relationships helps provide clearer explanations to patients, improves our knowledge of oocyte quality, and enhances rescue IVM ICSI outcomes. In our clinic, immature oocytes are placed into embryo slide with one-step media until the timing of ICSI procedure and the timing of rescue IVM procedure until we finished. We usually finish the ICSI procedure until 12 a.m. the day after oocyte retrieval. That concludes the presentation. If you have any questions, feel free to reach out, and I will be happy to answer them at a later time. Thank you very much for your kind attention.
Thank you, Dr. Takahashi, for your very interesting lecture. And of course, in the Q&A session, we would be very glad to come to different aspects of rescue IVM. But the initial question that comes up in my mind is that we would like to know what kind of light stimulation you use in your routine before getting such kind of diversity of egg maturation stages in your dishes per patient. So in your data, I saw that approximately somewhat less than half of the eggs are immature. And of course, this is linked to the time that you stimulate and also to the diameter of the follicles that are accessed by the clinicians. And so it can be very variable. I don't know if we need to spend a lot of time now to discuss, or rather move into the next question, into the next seminar. I think it is my pleasure to introduce Dr. Flor Sanchez. Flor Sanchez is our second speaker. She is a biologist that graduated from San Marcos National University, Lima, Peru, in 2012. She obtained her PhD degree at the Free University of Brussels under my guidance at the Follicle Biology Laboratory. So her research project focused on the refinement of oocyte in vitro culture conditions, firstly in the mouse model for the maturation of oocyte cumulus complexes, but also small follicles. And then she worked a lot on the promotion of the physiological environments and study it by gene expression of molecular markers of quality and differentiation, comparing in vitro cultured follicles and oocytes, and comparing it as a standard, as the golden standard, with in vivo produced cumulus oocyte complexes. At the end of her PhD, she was involved in a postdoc where she studied a new in vitro maturation culture system from small follicles that we call capacitation IVM. And so all this interesting work was done over a period of 5 to 7 years. And so it's my big pleasure to have Flor here. And please, Flor, take the floor and explain us what you did during your research period.
Okay. First of all, thanks for the nice introduction, Johan, and thank you to the organizers for the opportunity to be here. And it's really an honor and to participate of this great session about IVM. We have just heard in the first talk about rescue IVM, and this is a completely different story. I will talk about the status of traditional IVM, most specifically to the advancements that have been done over the traditional IVM protocol, and more specifically about kappa IVM. We'll see what is it. These are my disclosures. I'm a co-inventor of a patent on kappa IVM methodology, which is owned by the Free University Brussels. These are the contents of today. I will make a brief history about IVM and what is its status in the clinic now, variations in the IVM protocols, how we improve the efficiency of IVM or the standard IVM with kappa IVM, and the challenges, a bit of the challenges to clinical implementation and some concluding remarks. First of all, what is oocyte in vitro maturation? Well, it's an evolving assisted reproduction technique, which is defined as a maturation in vitro of immature cumulus oocyte complexes collected from antral follicles. This involves the transition from the prophase 1 to the metaphase 2 stage under controlled in vitro conditions. If you can see here, different to conventional IVF cycles where women receive many days of hormonal injections to make follicles grow until the preovulatory stage and then induce ovulation with an ovulatory trigger to obtain mature in vivo mature oocytes, and after that doing the ICSI or IVF. In IVM cycles, women receive a very minimal amount of injections, and now even they don't need even stimulation at all. So cumulus oocyte complexes are retrieved from small antral follicles. As you can see here, the range could be even from 2 to 10 millimeters, but mostly used in the standard IVM is 6 to 10 millimeters. And therefore, the immature cumulus oocyte complexes will undergo a period of culture so they can mature until the metaphase 2 stage, and then again, they will be fertilized by IVF or ICSI. And the idea of IVM is try to minimally treat the patient while we treat the gamete in vitro. So IVM is not a novel concept. It was first described in 1935 in rabbits, and it's a preferred technique in the livestock sector. The major advances have been done in basic research have been done in the animal field. However, the first initial contributions in human were done by Mencken and Brock, and most of this progress and the first definition of IVM was done by Dr. Edwards, who made significant progress by describing and establishing the kinetics and culture requirements for IVM oocytes in several mammalian species, including the human. After that, the evolution of IVM has been a bit slow. The first inclusion in the clinic, the introduction in the clinic was around the 90s. And later on, because these first attempts were not so efficient, then ovarian stimulation was introduced by use of ACG, FSH, or even a combination of both. But this led to some ambiguity regarding the IVM because it modified the literal meaning of in vitro maturation and created, let's say, different types of IVM. So these are regarding stimulation protocols, but also there were some strategies, but mainly at the laboratory level, so cultural strategies also with aiming to improve the efficiency of IVM. Specifically, I will talk about the biphasic IVM, which is kappa, but this started to be studied in around 2003, also by the group in Brussels under the guidance of Johan. And then we continue exploring this approach, and we got what we have now as kappa IVM protocol. But there are also other approaches that we can discuss further, which are interesting also and have been explored around 2005, for instance, a different culture, which is a three-dimensional IVM. It was done mainly in rescue GBO sites. And also more recently in 2023, an IVM that involves the co-culture of ovarian support cells, which are which is the first time they use induced pluripotent stem cells in co-culture with oocytes. And all of these strategies, what they aim is try to make an environment more physiological for the oocyte culture. Also, we have the introduction of IVM in the oncofertility field, which is very important from 2010 and 2020, and it continues. Okay, so despite this long history, it was not until 2021, that IVM was considered by the ASRM as a non-experimental technique. And the main indications was for PCOS patients who are at risk of ovarian hyperstimulation syndrome, but also patients with good ovarian reserve, meaning high antral follicle counts, and especially, I would say, very importantly, an alternative for fertility preservation in oncological patients who were not able to receive hormonal stimulation, or for which this was not indicated, especially patients with hormone-sensitive tumors. And because this is a treatment with less emotional burden and lower cost, it also offers a very nice alternative. And from the patient's perspective, these advantages even compensate the lower efficacy that currently has. However, this should be offered by centers with experience in this strategy. So it's not like a lack of necessity, but why this is not routinely used in the clinic then, despite this, all these advantages. And it's mainly because with the traditional protocols, we had lower effectiveness and success rates, and there's not enough long-term safety data. And also, and very importantly, is that there is a significant heterogeneity in the procedures, both clinical and laboratory protocols, which lead to ambiguities in the IVM definition that impact directly in obtaining all this data. However, recent studies with the biphasic IVM show encouraging results and are promising, and we could say that we could enter like in a new era to give a fresh start, IVM a fresh start. So these are the variations in the IVM protocol. We have the standard IVM, that's the ones that I defined at the beginning. It's a monophasic culture, so one-step culture, in which GBO sites will be mature until the metaphase 2 stage. It doesn't use ACG for the follicle for the patient stimulation. Then we have the biphasic IVM, which is a slight variation of this standard protocol by addition of one phase, which is called the pre-IVM phase, where immature GBO sites are arrested at the prophase 1 stage. And then in the second step, they will be induced to mature by in vitro maturation. As you can see here, all sites at retrieval are compacted, are cumulus enclosed, all sites, and you see the morphology is very compacted. Then we have the ACG prime IVF. And for this type of IVF, we can, as you can see here, at the moment of retrieval, following the ACG trigger, we can have like different types of cumulus oocyte complexes, where all sites can be at the immature stage. So they are the ones that will undergo, let's say, a real in vitro maturation. And there are also that are on their way of maturing, so in M1, and also those that have already matured at the moment of retrieval. And it's mentioned that, in fact, ACG prime IVM, it's a combination of in vivo mature and in vitro mature oocytes, because we have already some oocytes that mature in vivo. Then we have the rescue IVM that we have just heard in the first talk, and rescue IVM is indeed the maturation in vitro of cumulus denuded GB oocytes, or N1, as we have seen, which are collected from stimulated IVF exit cycles and have not responded to that stimulus to mature in vivo. Then we have another concept, which is the ex vivo IVM, or auto IVM. It's a maturation in vitro of immature GB intact COCs collected from ovarian tissue ex vivo and typically without any gonadotropin stimulation. And this strategy maximizes the fertility preservation potential of surgically removed ovaries. Okay, so in order to try to improve IVM efficiency, our team worked for many years trying to optimize the IVM, the standard IVM protocol, and that is how capacitation IVM born. Capacitation IVM, it's based on a biphasic IVM approach, has been inspired and founded on decades of animal research on key molecular processes regulating meiotic arrest and meiotic resumption, because knowing all this knowledge is crucial to improve in vitro maturation systems. So the principle of the kappa IVM strategy is that it aims to mimic in vivo follicular environment by using a physiological meiotic inhibitor during the pre-IVM stage that we call the kappa phase, the kappa step, which is C-natriuretic peptide. Then we use also a physiological meiotic stimulus, which is amphiregulin during the IVM phase. And the idea to have, or the aim to have this prematuration step is to promote cytoplasmic oocyte maturation and therefore developmental competence. And why is that? Because we are working with when we retrieve when follicles are retrieved from early antral follicles. And as you can see here, during oocyte development, oocyte gain meiotic and developmental competence in a gradual fashion, and meiotic competence is acquired earlier in development. This is the ability of oocytes to mature from the GB to metaphase stage, but developmental competence is acquired a bit later. And developmental competence is the ability of oocyte to form an embryo, to fertilize and produce a good quality embryo, and is associated with different processes, like for instance, the silencing of transcriptional activity, which is very high at the beginning, and it has to be switched off at the end of development. The accumulation of maternal RNAs that will be important during maturation and during the first stages of embryo development before the embryonic genome activates. And also the pattern of the chromatin remodeling, which is an epigenetic modification where the chromatin will pass from an uncondensed stage to a condensed stage. And all of these are associated with developmental competence. So one of the first and I think most important study we did in order to improve IVM, the IVM protocol, was to characterize the starting material that we were working with. So we did a characterization of the GB oocyte before and after IVM, looking for some cellular and molecular features, like, for instance, oocyte size, the distribution of mitochondria that has also been associated with developmental competence, transcriptional activity, and chromatin remodeling. And what we got is that we found that at this starting point, so at retrieval, we have a very heterogeneous population of oocytes enclosed in cumulus enclosed oocytes. Some of them which could be nuclearly and cytoplasmically immature according to these features, some that were nuclearly mature but cytoplasmically immature, and some others which were nuclearly and cytoplasmically mature. And then if we bring here also the concept of the pre-IVM, what we want with this phase, the capacitation phase, is try to make that this heterogeneous population is more homogeneous at the end of culture. So they are ready or more ready when they receive this meiotic stimulus. So we want them to reach this point, in fact. And one of the key points when we work with prematuration system is to maintain oocytes under meiotic arrest. And this is achieved by maintaining the levels of cyclic AMP very high within the oocytes. While we were doing these experiments trying to optimize the protocol, this publication came out. It was a publication of Meya Sang from EPIC's group, which is a very renowned team that work in oocyte development. It was in 2010, and they described for the first time how was the regulation of oocyte meiotic arrest in vivo, and they described this pathway, which is the CNP-NPR2 pathway. So CNP, the C-natriuretic peptide, is produced, is encoded by the MPPC gene, as you can see here, and is produced by mural granulosa cells. And this CNP will bind its receptor, the NPR2, which is a one in the cumulus cells, which is a guanylyl cyclase, and will produce cyclic GMP. And this cyclic GMP will diffuse into the oocytes and will inhibit the activity of the phosphodiesterase 3A, which is the one degrading the cyclic AMP within the oocyte. Therefore, when this impedes the degradation, the cyclic AMP levels will be maintained very high in the oocyte, and therefore the arrest will be maintained in the oocytes. So this is how it physiologically occurs, the meiotic arrest. And from that moment on, CMP was used by different groups to try to regulate meiotic arrest in vitro. In GB oocyte from preovulatory, mid-size, and also small antral follicles, mostly after hormone priming, as you can see here in different type of and different species. However, we chose to work with a more challenging model, and therefore this is the first publication by Sergio Romero, where we used unprimed juvenile mice and do the pre-IVM of these oocytes, and we were successful in maintaining oocyte meiotic arrest for 48 hours. And also the most important thing, I think, is to promote the oocyte cumulus communication. You will see why. And we could also promote the transition of the chromatin from the uncondensed to condensed stage. After IVM, we got a significant increase in the maturation rate and also significant increase in blast over 2 cell rate. And this is what I highlight as more important, because for us it's like key for the kappa IVM system to work. This is the maintenance of the transsonal projections, which connect cumulus cells and oocytes. And they, in fact, some years later, were also revealed or confirmed, confirmed to be functional by this nice study by Sao in 2020 from Gilchrist Group in Australia, where they also studied and reproduced this kappa IVM system, and they found that during the pre-maturation or kappa phase, the gap junctions remain open, and therefore during the first 24 hours that they studied, and therefore there is the oocyte and cumulus cells, there's a continuous communication between oocyte and cumulus cells. Later on, we did this translational study. So the first pilot study in PCOS patient that was in Brussels. We were also successfully in maintaining the meiotic arrest for 24 and 46 hours, and also promoting cumulus oocyte communication, as you can see here by the presence of transsonal projections, even when in a prolonged culture, 46 hours. And we could also, at some level, we could promote chromatin transition to the condensed stage, especially when we had a prolonged culture. After IVM, we got a significant increase in maturation rate in the kappa versus the standard protocol. And this could be reproduced after the validation studies were done in PCOS patients in Vietnam by the team of Vietnam, who now is the one leading all the clinical trials, or most of the clinical trials about kappa IVM. And we got also a significant increase in the amount of good and top quality embryos with this protocol compared to standard IVM. And this can be attributed to the kappa phase, but also we have to remember we have this important compound factor here, let's say, which is amphiregulin, that we use to induce meiotic resumption in vitro together with FSH. And we chose it because amphiregulin is the most abundant EGF-like factor found in the follicular fluid of primate and human. And its concentration have positively correlated with human oocyte developmental capacity. Here I show a nice graph from Conti and Franciosi, and Conti group has studied extensively mechanisms of oocyte in vivo maturation. And as you can see here, amphiregulin is here present. This is secreted by mural granulosa cells after LH binds its receptor. And amphiregulin diffuse through the follicular fluid and will bind its receptor, which is the EGF receptor, present in the cumulus cells, and it's the direct signal that the cumulus cells will see in vivo. What they proposed is that both amphiregulin and FSH have an essential role in the final acquisition of oocyte developmental competence during the periovulatory stage. And they propose they do that by enhancing or increasing the translation of selected maternal RNAs within the oocyte, which are involved in oocyte maturation, and also they remark the essential bidirectional communication between oocyte and cumulus cells, where oocytes produce and secrete factors that will influence the cumulus functionality, for instance, to promote cumulus mucification and expansion during IVM, during maturation. However, for this to work, this EGF receptor signaling has to be functional. And it's been described or reported by Ricciani and other authors that in small antral follicles, this pathway is not as mature as when it's in large antral follicles. And therefore, small follicles also show poor developmental competence compared to those from our large antral follicles. So what we believe is also that given this prematuration step, we also, by providing this intercommunication between cumulus and oocyte, we provide a period to promote the EGF receptor functionality in the cumulus cells, and therefore this improves the response to the EGF-like peptides. And this we have studied also in collaboration with the Vietnamese group, Brussels group and Vietnamese group, and we show this paper about the positive effect of amphiregulin on human oocyte maturation, where in a population of cumulus oocyte complexes that all of them undergo kappa, if you just use a different stimulus, one amphiregulin and the other a standard stimulus using conventional IVM, we see a significant effect of amphiregulin increasing the maturation rates. And although it was not significant, we could also see that pregnancy rates and live births were increased by number. About the clinical outcomes, this is the first publication showing the clinical outcomes in 2020, and we got that under kappa IVM protocol, we have a significantly higher clinical pregnancy rate per embryo transfer, and also a higher live birth rate per embryo transfer. And now, after many years of application, more than 1,000 children have born already in Vietnam specifically, but there are also some other few in other countries that have started to apply kappa IVM. And the updated live birth data up to now, it's that after first transfer of 2 day 3 embryos, live birth rate is from 35 to 47%, and after transfer of day 5 at day 5 blastocyst is 33 to 38%. Yeah. About the safety studies, when using kappa, we have analyzed blastocysts in the first studies by doing a PGTA and also by measuring the epigenetic profile, and we found that there's no differences compared to blastocysts obtained after conventional ovarian stimulation. About the pregnancies, there is also no different complication compared to IVF cycles. And the children follow-up also shows that there is no difference in the cognitive development and physical parameters compared to IVF and ICSI, some of them even compared to natural conceived children. So which patients can benefit of kappa IVM? Then we have just seen as a regular IVM, PCOS patients, women with high antral follicle count, normal responders also. It could also be beneficial using it in social freezing programs. But one of the most important things that I like also is the use in fertility preservation when it can be used in combination with auto IVM. And this is one first publication that from Kirillova that applied the kappa IVM together with auto IVM and showed an improved maturation competence of ovarian tissue oocytes by using it compared to the standard IVM protocol that they did in a sibling study. Why not also to think it's a strategy that can give hope to other type of syndromes like the ones gonadotropin resistant ovary syndrome and also patients with low ovarian reserve. Now about the challenges to the clinical implementation of IVM in general, we have here that in this publication we discuss a bit of that. From embryologist perspective, we have that in relation to the technical handling, there is some training to do that we can also discuss further later on. And however, the learning how to manage these oocyte, cumulus oocyte complexes from a small follicle require trainings which are not as challenging as those as learning ICSI or embryo biopsy. And there's important that there is some timing adjustments in the lab because also we need to, there is one extra phase, so not IVM, but it's a biphasic IVM, so you need one more day of culture. However, you don't need extra equipment in the lab. And from the clinician's perspective, there is some of the main challenges, I would say, is the retrieval of the small follicles. So the clinician have to pass a learning curve. Also, the use of some specialized needles to retrieve and access follicles which are very small. And specifically for kappa IVM and something that may influence its success, which is important to take in consideration when you want to implement the system, is the selection of COCs, because they need to be obtained as intact COCs, and this integrity needs to be maintained during culture, because the system works, I mean, the kappa works through the NPR2 system, CNP-NPR2 system that in fact communicates oocyte and cumulus cells. So therefore, if we have a partially denuded oocyte or even if it has less cells than this will impact the results. So it's key to obtain these ideal morphologies to try to obtain good quality oocytes and blastocysts. So trying to conclude, kappa IVM has been shown to be robust and reproducible IVM technology, showing consistently improved results in maturation rates and clinical outcomes. It has been proven to be valid and beneficial for PCOS patients. And now, because there is one publication that I couldn't mention before, but that revealed that there is no need of FSH stimulation. It enhances the efficiency of auto-IVM approach for fertility preservation, although this needs to be further explored. And its adoption of this strategy have been able to address safety concerns, and children follow-up so far is reassuring. So there are many things that still could be further explored with this system, like for instance, trying to refine cultural conditions for smallest follicles, thinking on the oncofertility field, also trying to refine conditions for the partially denuded COCs that we just mentioned. Why not to explore more the prolonged kappa culture, also in order to give more possibility to the very small follicles to attain developmental competence. Also to use in culture different factors like oocyte secreted factors, antioxidant to modulate oxygen tension, etc. And some of these have already been addressed partly, or in a good part, I would say, by the FOBI, Follicle Biology Lab in Brussels. And these are very nice publications from PhD students, which are now doctors, that have study extensively also capacitation IVM in mouse, and some of them also in the human model. So just to mention that this kappa IVM project is a project of international collaboration. It's led by Professor Johan Smits, and also a very large contribution of the Vietnamese group, like here, from Dr. Lan, and how. And in fact, this list should be extended because now there are more people motivated to use the system. And just to finalize, the road from this from basic science to the clinical application of kappa has not been fast, of course, no, but we have gone to basic research, translational research, validation studies, safety data, some key randomized trials, and also up to now there is some basic research done in mouse and even horse by a group in Ghent, which I like, and also some research in human IVM that is continuously being done. So with that, I will just end the presentation, and thank you everyone for your attention.
Thank you, Dr. Flor Sanchez, for this amazing presentation. So it's time to steer the discussion and move to the question and answer session. So please join and don't hesitate to turn on your cameras and participate. So I may start the first question that has been asked actually by several participants tonight about the IVM media. So you talked about mimicking the follicular environment, but we still don't know the exact composition of the media used. And could you consider in the future adding other environmental factors, such as other cells to culture with, or other biomaterials maybe?
Yes, thanks for the question. Indeed, the composition of the IVM, the kappa IVM, the compounds of this media as it is used now, it's described in different papers. We have described all the composition because it's not only to the use of CNP, which is physiological meiotic arrestor or AREC, but we also use factors like FSH, insulin, and estradiol, who also is involved in the CNP-MPR2 pathway. And these are also used in physiological concentrations. So this is also something that that we have studied for some years. And well, as I mentioned, it's described in different papers. I wish I could have put it here. But then, yes, of course, there is still, it's like when we talk among us, it's like the version 1, because we can still, of course, improve this composition if it will benefit outside developmental competence. The idea in the mouse, for instance, what it was used and also impact positively was the use of outside secreted factors that we could also try to include it for human cultures. And this is something that is still further studied in Brussels as research. However, yes, I think it's important also to consider other approaches that can be used in combination, like, for instance, why not the materials, as you mentioned, or trying to do a combination of approaches like even the three-dimensional that I just mentioned, that was never explored further, although in animal model it's been explored, but not in the human yet.
Thank you, Flor. And sorry, Christiane, I think I think a lot of people are asking, like, how is that utilized maybe also in rescue IVM? I think we've got questions from Maria Jose Escriba in Spain. My apologies for my pronunciations, as well as Alexander and Nora in the UK, and what sort of media was Cato Ladies Clinic utilizing? By the sounds of it, they're utilizing the same step 1 media that they would use for their regular mature oocyte, so no difference is our understanding. But I think a good question would be, should we be using the same media for rescue IVM that we're doing, with our mature oocytes, or should we potentially be utilizing some of the components or something maybe from true IVM? So maybe, I don't know if Dr. Land maybe can answer that, if you have any experience with rescue IVM, or if you think that perhaps we should be using some additional supplementation in our media for rescue IVM to maybe have slightly better results. What are your thoughts on that?
Yeah, hello. we primarily do Kappa IVM at our center. Yeah, sometimes we also have cycles with low maturation of oocytes in the stimulated cycles. So we perform rescue IVM, and for the media use, we just for rescue IVM, we just use the commercial IVM media available now in the markets.
Perfect. Thank you.
Dr. Lan, can you please explain a little bit what is the patient pathway to Kappa IVM? How these patients are chosen, how they are prepared, counseled?
For Kappa IVM, it is a kind of a standard procedure at our center for women with PCOS that have indications for ART. For the protocol, Kappa IVM protocol at our center, we don't use any gonadotropin. We don't use any HCG. And we allow patients to choose the day for oocyte pickup because no gonadotropins, no HCG, and they are PCOS women, so they have no cycles. So then any day is just okay for OPU, and we allow patients to choose any day that is most convenient for them. Then we perform OPU, and we culture the system. The culture system is similar to what Flor has just presented. I mean, 2 steps. The first step is Kappa step for 24 hours, and then the maturation step for 30 hours. And then we check for maturation, perform ICSI, and the rest of the procedure is very similar to the routine IVF. And with Kappa IVM, because at our center, we have been going through several different techniques of IVM. So we first started with HCG IVM. It means that we give patients a few days of FSH and stimulate and HCG 10,000 IU, and then we perform OPU. But with that technique, we could never go beyond day 3 embryos, and we found that the quality of embryo is not so good. And then we shift to standard IVM. Standard IVM means that a few days of FSH, no HCG, but just one phase culture system. And after that, we move to Kappa IVM. So I would like to emphasize that with the biphasic IVM, like 2 phases of IVM culture, Kappa and IVM, it allows I think the oocytes, the competence of oocyte is much better. So with Kappa IVM system right now, we don't use gonadotropin. We can grow the embryos to blastocyst stage, and currently we just transfer single blastocyst for women undergoing Kappa IVM. And recently we also perform fresh transfer in the Kappa IVM cycle. So it is totally, completely convenient for patients, convenient, safe, and effective. Yeah, I could say that Kappa IVM is really efficient and convenient for our patients.
Thank you.
You mentioned, I read from the questions from the floor that a lot of questions, of course, are coming regarding the safety of the procedure. And again, Lan, I'm sorry to address you. It's the middle of the night, but I mean, maybe you said already something about it, but what are the measures that you have taken? Of course, like Flor has explained, we analyzed the blastocysts and compared them for genetic and epigenetic markers, and this is all published it's in the literature. But you have done a remarkable work on the follow-up of the children. Maybe you can say something about that.
Yeah, thank you. Thank you for the opportunity to share about our studies on the safety of children. So we, in 2020, we did the first large RCT comparing kappa IVM and IVF in women with high androgen follicle count. And we found that the live birth rate was not inferior for the kappa IVM group as compared to the IVF group. So after the RCT, we follow the children born from that RCT until 2 years. And we assess the development of the children at 2 years using ASQ3 tools and reflex tools. And we found that there's no difference between the development of kappa IVM of children born after kappa IVM as compared to IVF. And also the physical growth of the babies were similar between the two groups of patients. And we also collected the biomaterials from babies born from kappa IVM, and we perform epigenetics variation comparing between babies born from kappa IVM and IVF, and we found no difference in the epigenetic variations between two groups of children. And currently, we are collecting data follow-up of 5 years after the RCT that had been done in 2020, and we are going to doing the analysis of the data on the safety of children at 5 years after the doing kappa IVM, and we are going to publish very soon.
Thank you.
Very reassuring too. I was going to say that would definitely be one of my questions as well. As a prior embryologist, would be, are there epigenetics that we're concerned about? Do you think we should be doing PGTA? I know that's not going to show us methylation changes and stuff like that, but is PGTA perhaps recommended for kappa IVM embryos? Is that something that would be helpful at all?
In Vietnam, we don't. Like PGTA is not routinely indicated for all kappa IVM patients. We perform PGTA for kappa IVL for women undergoing kappa IVM. But if there's indications, for example, the advanced female age or recurrent miscarriage, or for other indications like screening indications, but we don't routinely do PGTA.
Perfect. So the same as just our standard indications.
Yeah.
That's great.
Yeah.
I had one more question. Sorry to steal the stage for a second. Are there any handling considerations when working with the kappa IVM eggs, kind of again related to the epigenetics? Are they more delicate? Is there anything there that we need to consider if you're in the lab working with kappa IVM?
Yeah, I can say from the clinical side and from laboratory side, probably Flora can share with you her experience. From the clinical side, I think kappa IVM is a simple procedure, very simple and convenient. You don't need to follow, have like a comprehensive visit or comprehensive monitor patients. Yeah. For the OPU techniques, it is very similar to the routine IVF. Basically, there are two types of needles used for oocyte pickup in kappa IVM cycle. One system, one type is a double needle system, and the other type is a single needle system, like the routine IVF procedure. Why double needle system? Because you will know that the characteristics of the oocytes of the ovaries, the unstimulated ovaries. Usually the ovary is small, the follicle is very small, and especially the PCO, the polycystic ovaries, the tissue, the stromal tissue is thick and hard to puncture. So for the beginners, I think if you use the double needle system, it will help to increase the recovery rate. Basically, there are two needles. The double needle system, there are two needles. The external needles, 17 gauge, 30 centimeters. You puncture the outer needles first into the ovaries and fix the ovary. Just place the tip of the needles under the, very superficially, just under the cortex of the ovary. That one is used to fix the ovary, because the most difficult part of OPU in unstimulated ovary is the mobility of the ovaries. So the outer is to fix the ovary, and the inner go through the outer and puncture the small follicles and pick up the oocytes. And for the one that like very familiar with the OPU procedure, we can use the same types of needles with the IVF procedure. The pressure we use is the same.
Okay.
Yeah, very similar, but I think simple.
Thank you.
And
So,
about the laboratory part, well, in fact, yeah, there has been much concern about epigenetics, of this, or how would the influence at this level, because it's a long culture. I mean, it's a pre-IVM plus IVM. That's why we have studied since the beginning the blastocysts produced by IVM, and also the methylation patterns, and everything shows that these are comparable to conventional control ovarian stimulation. I mean, blastocysts from control ovarian stimulation cycles. However, yes, it's true that about handling it more carefully, I think that as I mentioned in one, I think the last slide, is because in general, all sites can be susceptible to different to its environment, so to different changes in this microenvironment that is growing in vitro. So I think one of the most important things is try to maintain, try to treat them delicately in the way that try to maintain all the time the integrity of cumulus cells and oocytes during culture and from the beginning, because there is nice study from one PhD student from the follicle biology laboratory about the transcriptional profile, and she compared even the transcriptional profile of these fully surrounded cumulus oocytes or the partially surrounded, and there is differences. So meaning that the fact that oocyte may not be fully surrounded by cumulus cells can impact also its development and also at this transcriptional profile level. However, this can be rescued by some alternative. For instance, she used the procumulin addition during culture, which restore this transcriptional profile. So there are ways still to explore, but this microenvironment is very delicate. So I would say that the addition of factors should be really like done in a specific way, you know. So it's not, we cannot, we try always to work using defined factors, so we know the direct effect that that can have in the oocyte and its development in every country.
Well, Doctor, sorry, Christiane,
ahead,
we're
go ahead.
so synchronized. This is why it worked during my PhD. So actually, I have a question about oncofertility. And you mentioned, Dr. Flor Sanchez, oncofertility, the application of IVM or kappa IVM for cancer patients. So how do you position today the IVM compared to, let's say, other alternatives for cancer patients, such as the transplantation of cryopreserved ovarian tissue, or even the transplant of artificial ovary, because I think it's still also a very promising alternative.
Yes, I think it's very valuable to study all approaches that can give opportunities for oncofertility patients. I think auto IVM is a really nice strategy because, for instance, it can be combined, as I mentioned, with the ovarian tissue cryopreservation, and you can have all this material all the time when you cryopreserve ovaries. So you just take advantage that the ovaries is there, and you learn to isolate these cumulosite complexes to put them in culture. And we believe that because the kappa is also providing this extra phase, it will also help these oocytes which come from a more underdeveloped status compared to those that, for instance, have received any stimulation or so. I think this is a very valuable procedure to explore further in this field. And I think it needs, of course, refinement of conditions. For instance, I would say that because they might be more underdeveloped, the extension of this kappa phase could be studied for these oncofertility cases. No? And also, one of the advantages is that if you culture them ex vivo, then there is, compared to the other strategies, you don't reintroduce tissue that may be, for instance, still with some residual cancerous cells, if there would be some risk there. No, that's what I would say.
That's a valuable point.
One last question because we are running out of time. Dr. Sanchez, in settings where conventional stimulation is feasible, how does kappa IVM performance compare, especially regarding cumulative live birth rates and cost and even patient preference?
Would you like to reply, please, Dr. Lan? I think.
Yeah.
Thanks.
It's in conditions where we can offer conventional stimulation. How can we compare with the kappa IVM in terms of performance?
Yeah. So at our center, we integrate IVM into our existing IVF system. At our center, PCOS women who have indications for ART, for example, they have male factor. The couples have male factor that requires doing ICSI. So instead of doing stimulation, we put the women on kappa IVM. And regarding the performance, the convenience, we just don't do stimulation at all. We collect oocytes. So I think regarding the patient preference, most patients especially women with PCOS, they prefer kappa IVM rather than stimulation. And when they come to the center, they first request kappa IVM. They don't want to do a stimulation. Regarding the effectiveness, the live birth rate per transfer is similar between IVM, kappa IVM, and IVF. However, because the total number of embryos of kappa IVM is a little bit lower than that of IVF, so the cumulative live birth rate of kappa IVM is lower than IVF, the stimulation IVF. Regarding the cost, kappa IVM, the cost of kappa IVM is only about 2/3 of the stimulation IVF cycle.
Do you know what the difference in live birth is cumulatively? Because I'm thinking like if you undergo one cycle and it's significantly cheaper, but if you have to undergo 2 cycles to get a similar live birth cumulatively, just curious if you know what the difference is.
Actually, with kappa IVM, we could manage to generate about a mean of 4 embryos. And for 4 embryos, actually that is most of the time that is enough to for a family. So yeah, especially the success rate, I mean like the live birth rate per transfer is similar to IVF. So we don't see like many patients that they have to repeat up to 3 IVM cycles. Yeah.
I think we have a lot of questions still, and the night is short. So at least here, it's still night. So thank you everyone for joining us. This is the end of our session today that was sponsored by Future Fertility. And I would like also to thank all our co-hosts. So we have Professor Christiane Amorim, Professor Johan Smits, Dr. Lan Vrong, and Dr. Julian Fjellstad. Sorry if I mispronounced your name, but keep pushing the boundaries of IVM, and see you on the next session. Dr. Yuan, you have a closing word to say about the next session?
I thought it was a fantastic session. I thank the organizers for having the possibility to discuss IVM. I think it's still in evolution. I think we have a very good platform, and I think Dr. Lan's arguments are, I think, very convincing to all of us. And I hope that all that are in the meeting here are triggered also and participate, because any improvement that will come should come from good ideas that can be brought by all of you. And just to close, I think we have now an after-party session for those that still want to discuss. I think there is a link that is provided by the organizers for that. And my last word is, please join the I3 team next week for the next great webinar, which is organized with RBMO editors, reproductive biology online editors, and close collaborators. And it's about writing and submitting manuscripts, a masterclass about it. It's very important because, yeah, it's publish or perish. So please all join next week's webinar. Thank you.
Thank you,
Thank
all
you.
of you. It has been a great pleasure, and I'd like also to thank the team from I3, the organizing committee, as Johan already mentioned. But I'd like to name the organizers: Jacques Cohen, Peter Nagy, Mary Ann Cervantes, I hope I pronounced correctly, Thomas Elliott, John Farley, Violetta Sora, and Giles Palmer. Thank you, all of you, for being here with us today.
Thank you.
Thanks.
Bye.
Bye-bye.
Bye.
Thank you. Bye.